these chylomicrons aren't properly transporting fats. If we take a look at how chylomicrons normally form, we can better understand why sufferers of CMRD have trouble digesting fats. Chylomicron synthesis begins in the enterocytes of your small intestines. These are the same cells we looked at in the young girl's biopsy. Remember apolipoproteins? These were the decorative elements on our hot cocoa bomb. Apolipoprotein B48 is the most important structural element of a chylomicron. It's crucial to metabolism because it also guides lipoprotein formation and can control metabolic enzymes. After its synthesis in the rough ER, ApoB48 moves to the smooth ER and is readily degraded, unless phospholipids, cholesterol, and triglycerides are available. In the rate-limiting step of chylomicron formation, a microsomal triglyceride transfer protein combines reformed triglycerides from free fatty acids and monoacyl glycerols with phospholipids and cholesterol. At this point, the structure is still in the ER and is referred to as a pre-chylomicron. To reach a form that is useful for transport around your body, the pre-chylomicron must travel to the Golgi body for maturation. To consider why, we can think of this whole process like a toy assembly line in Santa's village. Santa's village receives all the parts, where the elves will assemble them into magnificent toys. Even though the gifts are made, they need to be passed on so they can be packaged into Santa's sash. That way they can be sent out to the world and distributed. If this is analogous to chylomicron formation, what might cause CMRD? If you sit at the assembly point, that's actually a different disease called abeta-lipoproteinemia, caused by a lack of ApoB48. CMRD stops successful transport to the packaging step. This is hypolipoproteinemia. How does this transport take place? It starts with the pre-chylomicron budding out from the ER into a pre-chylomicron transport vesicle. This is the rate-limiting step of intestinal escape. Early evidence suggested this ER exit was facilitated by COP2, which makes sense because this protein coat contains SAR1b. Recall that the SAR1b gene is implicated in CMRD. However, this mechanism of COP2-mediated budding is used for smaller particles, and chylomicrons are much too big. Siddiqui and colleagues showed that COP2 and by extension SAR1b are not actually needed for ER exit. They labeled rat small intestine mucosal cells. If budded, the pre-chylomicron transport vesicles were labeled. These were separated by sucrose density centrifugation. They found that ATP was required. More relevantly, COP2 proteins were associated with budding. But doesn't this support the old way of thinking? Actually, when antibodies against these proteins were added, budding still occurred, but protein vesicles that weren't transporting pre-chylomicrons were inhibited. This is how pre-chylomicron budding from the ER is thought to occur. A fatty acid binding protein, represented by FABP1 in the image, is used to facilitate the budding. Since the fatty acid binding protein can't bind directly from the cytosol, and ATP is known to be used upon ER exit, the process is thought to be regulated by a protein kinase C. ATP activates the protein kinase C and causes the phosphorylation of SAR1b. The result is that the complex dissociates, and the fatty acid binding protein is available to bind. SAR1b and COP2 are therefore not needed for ER exit, but may be implicated in the regulation of PCTV. The Siddiqui study supports this model, and also indicated that PCTVs would not fuse with the Golgi body in the absence of SAR1b. This suggests the SAR1b mutation of CMRD would affect entry into the Golgi body and chylomicron maturation. If this transportation is halted, the chylomicrons won't carry triacylglycerols to the rest of the body to create energy from their glycerols and fatty acids. The regulation of Golgi fusion is not well understood, so more research is needed to provide insight into what happens between the ER and Golgi body and when. For example, could a pre-chylomicron transport vesicle get released even if it won't be taken up by the Golgi body? If so, does it go back to the ER when it's not taken up? Some promising research indicates the use of snares in Golgi fusion. Researchers labeled pre-chylomicron transport vesicles and took advantage of the fact that a certain apolipoprotein gets added onto the vesicle only upon Golgi fusion. If ApoAI is not added, the vesicle has only been docked. Researchers performed a sucrose step gradient to separate out their desired products. This technique works according to the characteristic of density. The team used labeled triacylglycerols, which would be found within PCTVs, and incubated them with Golgi. Any pre-chylomicrons that had fused obtained ApoAI and became isodense with the Golgi. The study showed that by adding antibodies for several of the predicted snare components, PCTV Golgi fusion did not occur. An immunoblot or Western blot analysis showed that when the components were not boiled, those predicted to be part of a snare complex all revealed bands at 112 kilodaltons. By showing that the snare components had bands present at the same distance, in combination with the fact that they did not dissociate in the SDS page that was used in running the test. In other words, the components made up a single structure. The researchers could indeed that a snare complex, including these components, might be used in vesicle fusion of PCTVs. So far, what we've learned makes sense of the intestinal fat droplets we saw in the biopsies. If our dietary fats can't get properly packaged in the Golgi body, they can't be sent out to the rest of the body, and therefore will get stuck in the intestine. What about the vitamin E deficiency associated with chylomicron retention disease? Vitamins E, D, K, and A are actually all low in the case of CMRD. This is because they're the lipid-soluble vitamins, so they have to be carried around in the body by chylomicrons. Recall that chylomicrons are used in the first place because dietary fats avoid the aqueous environment of the cell. The low levels of these vitamins account for the ophthalmic issues associated with CMRD. Supplementation is sufficient to avoid these eye problems, but I predict that that would not be the same case for the related condition called A-beta lipoproteinemia, which we saw was due to a lack of ApoB48. I predict this because vitamin supplementation would not be sufficient if your body cannot process those vitamins at all. It might be tempting to think that any neurological conditions associated with CMRD are due to a lack of energy because your body can't transport dietary fats. Your brain's main source of energy is carbohydrates, but you do need these vitamins to help your brain. Therefore, it is again a lack of lipid-soluble vitamins that causes the neurological symptoms of CMRD. In terms of energy, however, a failure to thrive and stunted height and weight growth are associated with a lack of energy. Low cholesterol in CMRD might make you think, isn't cholesterol in those chylomicrons that are getting stuck and built up in the intestines? Wow, that's true. It turns out that cholesterol has to be measured in the blood. There it has an important role in adding into the structures of membranes. It also is used to carry around fatty acids in the form of high-density HDL or low-density LDL structures. We can quickly walk through how structures of various fat to cholesterol ratios end up in the blood. When a chylomicron successfully leaves the small intestine, as occurs in a healthy body, an apolipoprotein can bind onto a lipoprotein lipase, causing the triacylglycerols to go into the body cell. It separates into glycerol and fatty acids, which can both go on to make energy or be stored. What's left of that chylomicron, called the remnant, goes on to the liver. The remnant gets separated into its phospholipids, triacylglycerols, and cholesterol, and then reformed into VLDL, which is very low-density lipoproteins. The remnant gets separated into its phospholipids, cholesterol, and triacylglycerols, then reformed into VLDL, which is very low-density lipoprotein. VLDL again goes on to the lipoprotein lipase of a body cell, and what's left this time is an intermediate density lipoprotein. The similar process continues as the relative amount of cholesterol in the lipoproteins increases compared to the triacylglycerols, and the size of the particles decrease. Through this process, cholesterols are getting carried through the blood, where they can be measured. But in the abnormal case of CMRD, the chylomicrons never leave the small intestine. Because these high cholesterol lipoproteins are dependent upon the action of chylomicrons, CMRD does not allow cholesterol to be found in high levels in the blood. We can also see here that if a chylomicron is getting trapped in the intestine as it does in CMRD, it's not going to reach a body cell, and therefore the triacylglycerols within it will not be broken up into glycerol and fatty acids and will not be able to be used for energy or storage. This leads to the failure to thrive symptoms. So what do we know so far about chylomicron retention disease?
Run.
Overall, the mechanisms that are described in this video for chylomicron transport and formation are simply predictions supported by evidence, and more research is needed to fully understand how the process works and how CMRD might be cured. Consider for a moment that so much research is going into a disease that has only ever been diagnosed in 50 people. So much research is being done on such a rare disease because the findings actually have implications for the controversially named obesity pandemic, as well as the understanding of lipid-soluble drug metabolism. Research in that area is vitally important because fat is so widely abundant in food. In fact, food industries purposely combine fats with carbohydrates in order to make their food more appealing due to the biological drive for both sugars and long-lasting energy sources, fats, which are not naturally found together unless prepared by humans. You may have seen that one of the points that's not well understood is why chylomicron sizes are determined. Knowing that they're too large to pass through the Golgi body simply, future researchers could consider regulation of lipoprotein sizes to see if there's a way to get chylomicrons into the Golgi body. Understanding what can go wrong in fat metabolism is very important, so I would like to thank you for joining me on this learning journey, and I hope you have grown to appreciate all the work your body does just to metabolize one macromolecule. Thank you.